Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa. In today's video, we're going to take a look at the regulation of gene expression. There are so many genes in the genome, but not all of them are needed to be expressed at the same time or all the time. So how does the cell regulate that? Join me as we unveil those secrets. Prokaryotes and eukaryotes can regulate their gene expression in response to environmental conditions. In multicellular eukaryotes, gene expression regulates development and is responsible for differences in cell types. RNA molecules play many roles in regulating gene expression in eukaryotes. Bacteria can often respond to environmental change by regulating transcription, and this is something that we'll be talking about throughout this lecture. Natural selection has actually favored bacteria that produce only the products needed by that cell. This is a way for the bacteria to utilize the resources that are available and then not work so hard to express other genes that are not needed at that point in time. A cell can regulate the production of enzymes by feedback inhibition or by gene regulation. One mechanism for control of gene expression in bacteria is the operon model. Let's take a look at how bacteria can conserve resources by this metabolic pathway that also has feedback inhibition. So bacteria, if they are in a tryptophan-rich area, they do not need to make tryptophan because they can obtain it from their environment. However, in times where tryptophan levels are low in their environment or non-existent, they are able to synthesize it. And so what you see in front of you is a metabolic pathway that involves several enzymes, and this metabolic pathway is used by bacteria to make tryptophan. Tryptophan is an amino acid, and it's needed in order to produce proteins. So without it, the bacteria would not be able to produce proteins. So it always needs tryptophan either from its environment or it needs to be able to make it. When this metabolic pathway is turned on, these different enzymes can be synthesized by the genes that encode them, which means the genes then need to go through transcription to be made into mRNA and then translated into protein to make the different enzymes. When this metabolic pathway is turned on, tryptophan is made so that the bacteria can utilize that amino acid in making proteins needed for other things. Now, once tryptophan levels increase to a high enough level for the bacteria to be able to synthesize its proteins and reproduce, etc., tryptophan can negatively feedback on both sides of this metabolic pathway, meaning tryptophan can work in feedback inhibition by negatively feeding back and blocking the enzyme action of enzyme number 1. And this will stop the entire pathway from synthesizing tryptophan. Tryptophan also has the ability to negatively feedback on the regulation of gene expression. So if it stops gene expression, we won't get those enzymes synthesized, and if those enzymes aren't synthesized, then the pathway cannot proceed. So when bacteria is in an environment with low levels of tryptophan, we can turn on this metabolic pathway, increasing tryptophan being made and produced by the bacteria. And once those levels get high enough or the environment changes so that there's more tryptophan in the environment, then tryptophan itself can negatively feedback on the system and stop the production of tryptophan. This allows the bacteria to conserve resources and energy by only making tryptophan when tryptophan is needed. Now, the genes needed in order for bacteria to synthesize that amino acid tryptophan are all located together on the bacterial chromosome. That cluster of genes, all located together, is called an operon. So, an operon is a cluster of functionally related genes that can be coordinately controlled by a single on-off switch. That switch is a segment of DNA that is called an operator, and it is usually positioned within the promoter. So, an operon includes the operator, the promoter, and the genes that they control. The figure in front of you shows the basic structure of an operon. So we can see in an operon there is a cluster of different genes that are all functionally related. They're going to work together to do something. At the beginning of the operon, we see a promoter, and within the promoter region, there is an area called the operator. So this operator is going to be located usually within the promoter. And so this area of the promoter and the operator lies right in front of the actual genes within that operon. The other unique thing about the operon is that when the genes are transcribed by RNA polymerase, they are transcribed as one piece of mRNA, and then they are processed into different proteins after that. The operon can be switched off by a protein repressor. The repressor is going to prevent gene transcription by binding to the operator, that area right in front of the genes, and blocking RNA polymerase from doing its job. The repressor is the product of a separate regulatory gene. The repressor can be in an active form or inactive form depending on the presence of other molecules. So the take home is that on that operon, there is that region that we just looked at called the operator, where a repressor can bind, and if a repressor does bind, it can block RNA polymerase from doing its job, repressing the operon. So the operon would not be able to be transcribed or translated, meaning no protein would come out of that operon. So this is the unique thing about these operons, is that all the genes needed to do a single function are on this operon, and they can all be turned on or off together. A co-repressor is a molecule that cooperates with a repressor protein to switch an operon off. For example, E. coli can synthesize the amino acid tryptophan when it has insufficient tryptophan, as we've just talked about. By default, that trip operon is on. Now, the trip operon is the name given to that tryptophan operon that is going to produce tryptophan. So by default, that operon is going to be on, and the genes for tryptophan synthesis will be transcribed. When tryptophan is present, it is going to bind to the trip repressor protein, which is going to turn the operon off. The repressor is only active in the presence of its co-repressor, which in this case is tryptophan itself. Therefore, the trip operon is turned off if tryptophan levels are high. And so when tryptophan is present, it also has that ability to act as a co-repressor, binding to the trip repressor protein. Then at that point, it is going to repress the trip operon, and no genes are made. Because if tryptophan is present, then tryptophan does not need to be synthesized, and this allows the operon to be shut off or repressed. A repressible operon is one that is usually on, and binding of a repressor to the operator shuts it off. The trip operon is a repressible operon, as we've seen. So normally the trip operon would be on and genes would be able to be expressed, and it would be turned off once the inactive repressor was activated with a co-repressor. It would be turned on, that repressor would be turned on, allowing it to bind to the operator position on the operon, blocking the RNA polymerase. And in this case, the genes would not be expressed. So it is a repressible operon. It is usually on, and it can be turned off by the binding of a repressor. Let's take a look at the trip operon as a whole and talk about all the things that we've learned about the trip operon. Now, the trip operon is a repressible operon, meaning it is usually on and it needs to bind to a repressor to bind to that operator to shut it off. So here we see the trip operon in its entirety. It includes the promoter region, which has the operator within it. And then it also has the genes of the operon downstream of that. So RNA polymerase can bind to this normally and allow the genes to be transcribed into mRNA and then translated into proteins, those different enzymes that are needed in order for tryptophan to be synthesized. So this operon is usually on, allowing this to happen, where we get that mRNA, we get those different enzymes we talked about. Those enzymes are going to work in allowing for tryptophan to be synthesized. So as long as it's on, tryptophan is going to be synthesized, and this operon is a repressible operon, so it is normally on. The other thing that we see is this regulatory gene that does get transcribed into mRNA and then protein, and this is where we get the repressor. So the repressor comes from this regulatory gene that is not part of the operon. It is upstream of the operon, separate from the operon, and it is made, but it is inactive. It is in an inactive stage. Now, as long as tryptophan is absent, this operon is going to be working. When tryptophan is present, it is a co-repressor, and it binds to this regulatory protein or the repressor. It's going to bind to it, and what that does, it allows that protein to turn into its active form. So now it is an active repressor, and it can now bind to the operator. It fits into the operator, it binds to the operator, and when that happens, RNA polymerase gets blocked and the genes cannot be transcribed. So this operon gene synthesis is shut off. So gene expression is shut off by this repressor binding to that operator. An inducible operon is one that is usually off. A molecule called an inducer inactivates the repressor and turns on transcription. So this figure is an example of an inducible operon. So an inducible operon, its normal state is that it is normally off, and this is because a repressor is going to be binding to the operator, keeping the genes from expressing. So in this case, the genes are not expressed. They cannot be expressed because the repressor is bound to the operator. And this is the normal state of an inducible operon. However, a molecule called an inducer can bind to the repressor. This inactivates the repressor because now the repressor can no longer bind to the operator. And if the repressor cannot bind to the operator, then the genes can be expressed. So this is an example of the operon being induced, where that inducer binding to the repressor keeps it from binding, so it cannot bind to the operator region, allowing RNA polymerase to then express those genes. A different operon, called the lac operon, is an inducible operon, and it contains genes that codes for enzymes used in the hydrolysis and metabolism of lactose. A regulatory gene, LAC-L, located outside the operon encodes a repressor protein that can switch off the operon. By itself, the Lac repressor is active and switches the Lac operon off, placing this inducible operon normally in the off position. A molecule called an inducer inactivates the repressor to turn the Lac operon on. In the case of the Lac operon, the inducer is allolactose. This is an isomer of lactose. So allolactose binds to the repressor protein and it alters its shape so that the repressor can no longer bind to the operator sequence. So whenever lactose is present, allolactose will also be present. And so therefore, in the presence of lactose, the Lac operon can be turned on because allolactose will bind to the repressor, and then that allows for the repressor not to be able to bind to the operator anymore. Let's take a closer look at the Lac operon and how it works. Before, we were talking about the Tryp operon, and the purpose of the Tryp operon was to make tryptophan. So when tryptophan levels actually got high, it could negatively feedback on this system. And so the Tryp operon was needed when tryptophan levels were low. Now, what is the purpose of the Lac operon as we move into talking about the Lac operon, which is an inducible operon? So the Lac operon contains genes that are going to code for enzymes that are necessary in the hydrolysis and metabolism of lactose. That means it's going to help to break down that lactose so that the bacteria can actually utilize that lactose. So that's what these genes in the Lac operon are doing. So it makes sense that the bacteria would only want this operon on in the presence of lactose because that's when it's going to need these different enzymes. So the Lac operon is an inducible operon, meaning that it is normally off because the repressor, when it is made by this LAC-L gene, when the repressor is made, it is active, okay? So it starts out in its active form, and then it binds to the operator, and it inhibits the synthesis of any of the genes in this operon because RNA polymerase, which binds to the promoter, cannot proceed forward because this repressor is binding to the operator. So in this inducible operon, it is normally off because that repressor is binding to the operator. However, in the presence of lactose, that means allolactose will also be there. And allolactose is referred to as an inducer because when it binds to that active repressor, it inactivates that repressor. And by inactivating the repressor, now the repressor can no longer bind and the operator is opened. This allows RNA polymerase to continue to move forward and transcribe those genes into mRNA and then into protein. And now we get those enzymes that are necessary for the hydrolysis and metabolism of lactose. Inducible enzymes usually function in catabolic pathways. Their synthesis is induced by a chemical signal. So catabolic pathways are those pathways that break things down. Repressible enzymes usually function in anabolic pathways. Their synthesis is repressed by high levels of the end product because anabolic pathways are going to build things up or make that end product. Regulation of the Tryp and Lac operons involves negative control of genes because operons are going to be switched off by the active form of the repressor. Some operons are also subject to positive control through a stimulatory protein such as cyclic AMP receptor protein, abbreviated CRP. This protein is an activator of transcription. When glucose, which is a preferred food source of E. coli, is scarce, CRP is activated by binding with cyclic AMP, abbreviated CAMP. Activated CRP attaches to the promoter of the Lac operon and increases the affinity of RNA polymerase. This accelerates transcription. When glucose levels increase, CRP detaches from the Lac operon and transcription returns to a normal rate. The ability to catalyze compounds like lactose enables the cells deprived of glucose to survive. So what's actually happening here is that activated CRP can attach to the promoter of the Lac operon and allows it to increase transcription. But when glucose levels increase, it detaches that CRP and allows transcription of the Lac operon to return to a normal rate. And the reason is because bacteria would rather use glucose if glucose is present in order to use glucose for its energy, and use lactose only if glucose is absent. Let's take a look at how this works looking at the Lac operon again. So this is the Lac operon that we just talked about before, and this first part of the figure shows us that lactose is present and glucose is scarce, okay? So what is going to happen in this situation if lactose is present, like we just talked about before, that allolactose is going to bind to that repressor that is normally active, and it's going to inactivate it. By inactivating it, that's going to allow the RNA polymerase to bind to the promoter and transcribe those genes into mRNA, and then they can be translated into protein. The other thing that we can see happening if glucose is scarce is that cyclic AMP can bind to the CRP, and that active component of both of them being together is going to bind right before the RNA polymerase. What this does, it just stimulates transcription to happen even more. So it's going to be a positive regulator promoting more transcription to happen. Now, as I just mentioned, the bacteria would prefer to use glucose if glucose is there, but this allows the bacteria to also utilize lactose if glucose, for whatever reason, isn't present. So if we look at the bottom portion of this, if lactose is present and glucose is present, then the bacteria are going to prefer to use glucose. And what happens is when glucose is present, that cyclic AMP level is low. And if that cyclic AMP level is low, it doesn't allow this to happen where cyclic AMP binds to the CRP. So it keeps CRP inactive without the cyclic AMP binding to it. And that's because when glucose is present, those CAMP levels are just low and they can't bind, and this cannot bind to the CRP binding site. And if that's the case, then that is going to bring transcription levels back to normal. Lactose would still be present, so allolactose can bind to the repressor, keeping it inactivated. But because this CRP is not activated in binding, RNA polymerase is less likely to bind, and so gene expression will be pretty low in this case, allowing the bacteria to favor using glucose as its energy form. What about eukaryotic genes? Eukaryotic gene expression is regulated at many stages. All organisms must regulate which genes are expressed at any given time. Genes are turned on and off in response to signals from their external and internal environments. In multicellular organisms, regulation of gene expression is essential for cell specialization. Almost all the cells in an organism are genetically identical because they're going to have the same exact genome. But there are different cells that make up an organism, which means there has to be some differentiation so that those cells can actually be specialized cells, like the difference between a skin cell and a neuron. So differences between cell types are going to result from differential gene expression, which is the expression of different genes by cells, even though they have the same genome. Abnormalities in gene expression can lead to diseases, including cancer. Gene expression is then regulated at many stages. This figure shows the stages in gene expression that can be regulated in eukaryotic cells. So again, as we've mentioned, there are many areas in where gene expression is regulated. We see chromatin modification, DNA unpacking. So at the DNA level, there is gene expression regulation. At the RNA level, there's also gene expression regulation that we see a transcription, RNA processing, and the transport to the cytoplasm. So mRNA is made in the nucleus and then it still has to be transported to the cytoplasm. That process is also regulated. mRNA can be degraded as well. And then we move into the protein part where gene expression is still regulated at several stages in translation, in protein processing, in transport to its cellular destination, and in the degradation of protein. So there are many stages that are regulated in gene expression to make sure that the proper proteins are being made in the cell. Regulation at the DNA level is regulated by the chromatin structure. So that structural organization of the chromatin actually helps to regulate gene expression. Genes that are within the highly packed heterochromatin are usually not expressed, and in euchromatin, gene transcription is affected by the location of the nucleosomes along the promoter and the sites where DNA attaches to the protein scaffolding of the chromosome. Chromatin structure and gene expression can be influenced by chemical modifications of the histone proteins of the nucleosome. These modifications are catalyzed by specific enzymes. In histone acetylation, acetyl groups are attached to an amino acid in a histone tail. This process opens up the chromatin structure and allows for the initiation of transcription to happen. The addition of methyl groups or methylation can actually condense chromatin and reduce transcription. So by adding these modifications, we can either open up the DNA and allow for transcription by acetylation, or we can reduce transcription by methylation. And different enzymes will be able to come in and do these different things. We can see that the histones have these tails that extend out from them. The DNA wraps around it, and the histone, along with the DNA, is referred to as the nucleosome. And normally, when they're unacylated, those histone tails, the DNA is very wound and you can't get to it. However, when acetylation happens, then that allows the DNA to open up and it's more accessible for transcription to happen. On the other hand, DNA methylation, which is the addition of methyl groups to certain bases in DNA, is associated with reduced transcription. DNA methylation can cause long-term inactivation of genes in cellular differentiation. So if the cell wants to turn off certain genes, those those pieces of DNA can be methylated. In genomic imprinting, methylation regulates gene expression of either the maternal or paternal alleles of certain genes at the start of development. So again, methylation is a way that the cell can reduce transcription of specific genes so that they're not always being transcribed. Although the chromatin modifications just discussed do not alter the DNA sequence itself, it's just modifying the DNA so that either it can be transcribed or not transcribed, but it is not changing the actual sequence. These modifications can be passed to future generations of cells. The inheritance of traits transmitted by mechanisms not directly involving the nucleotide sequence is called epigenetic inheritance. Epigenetic variations might explain cases where one identical twin develops a genetically based disease while the other does not. Chromatin modifying enzymes provide initial control of gene expression by making a region of DNA either more or less able to bind the transcription machinery. Associated with most eukaryotic genes are multiple control elements, segments of non-coding DNA that serve as binding sites for transcription factors that help regulate transcription. Control elements and the transcription factors they bind are critical to the precise regulation of gene expression in different cell types. To initiate transcription, eukaryotic RNA polymerase, remember RNA polymerase is what's going to be used for transcription. Transcription is going to go from DNA to mRNA. So eukaryotic RNA polymerase is going to require the assistance of transcription factors. General transcription factors are essential for the transcription of all protein coding genes. In eukaryotes, high levels of transcription of particular genes depend on control elements interacting with specific transcription factors. Proximal control elements are located close to the promoter, and distal control elements, groupings of which are called enhancers, may be far away from a gene or even located in an intron. Each enhancer is generally associated with only one gene and no other. An activator is a protein that binds to an enhancer and stimulates the transcription of a gene. Activators have two domains, one that binds to the DNA and the second that activates transcription. Bound activators facilitate a sequence of protein-protein interactions that result in the transcription of a given gene. The currently accepted model suggests that protein-mediated bending of the DNA brings the bound activators into contact with a group of mediator proteins. The mediator proteins interact with the general transcription factors at the promoter. This helps assemble and position what is called the pre-initiation complex. Here we can see in the distal control element, we have the enhancers, and the activators can bind to this area where the enhancers are. And then what happens is we have a group of mediator proteins that come into this area, and other proteins such as the DNA bending protein that allows the DNA to basically loop over and bend. And this allows that area where the enhancers are to be able to interact with general transcription factors and mediator proteins in order to allow for transcription to happen. So all these proteins are going to be needed in order basically for the RNA polymerase to bind. And when the RNA polymerase can bind, then and only then can transcription happen, can that RNA synthesis happen, only when the RNA polymerase binds. And RNA polymerase needs all these other factors and proteins to be able to know where it needs to bind on the DNA. Some transcription factors function as repressors, inhibiting expression of a particular gene by a variety of methods. Some repressors can bind directly to control elements and block activator binding. Others interfere with the activators themselves, so those activators cannot bind the DNA. Some activators and repressors may indirectly affect transcription by altering the structure of the chromatin. A particular combination of control elements can activate transcription only when the appropriate activator proteins are present. In this figure, we can see that happening where the appropriate activator proteins are available up here to allow for the binding of all the other proteins that are necessary for RNA polymerase to bind and initiate transcription. But when the appropriate available activators are not the ones that bind to another type of gene in the activator enhancer area, then that gene will not be expressed, okay? So the activators need to be present in order to bind, to allow for the other proteins to bind, creating this pre-initiation complex where RNA polymerase can bind and allow for gene expression. If those activators are not there in these two cases, then we cannot get transcription of the gene. Co-expressed eukaryotic genes are not organized in operons like we've seen in prokaryotes. There are a few minor exceptions. These genes can be scattered over different chromosomes, but each has the same combination of control elements. Activator proteins in the nucleus recognize specific control elements and promote simultaneous transcription of the genes. Transcription alone does not account for gene expression. Regulatory mechanisms can operate at various stages after transcription has happened. Such mechanisms allow a cell to fine-tune gene expression rapidly in response to environmental changes. In alternative RNA splicing, different mRNA molecules are produced from the same primary transcript depending on which RNA segments are treated as exons and which as introns. It is a proposed explanation for the surprisingly low number of genes in the human genome. Actually, more than 90% of the human protein coding genes undergo alternative splicing. What does that mean? So this is a proposed explanation for how there's a little low number of genes and then a lot of different proteins. And what happens here is that the DNA is transcribed into RNA, and then that RNA can go through alternative RNA splicing, creating different mRNAs. And those mRNAs are then going to be converted into protein. So based on the different exons that are here, the exons are going to be put together, the introns get taken away, and the exons actually have the ability to be put in different ways. So this is just an example where in this one, when it was spliced, 1, 2, 3, and 5 were put together. In this next one, 1, 2, 4, and 5, and they are going to give rise to different proteins, still arising from the same DNA sequence, but allowing for different proteins to be made. The initiation of translation of selected mRNAs can be blocked by regulatory proteins that bind to sequences or structures of the mRNA. Alternatively, translation of all mRNAs in a cell may be regulated simultaneously. For example, translation initiation factors are simultaneously activated in an egg following fertilization. The lifespan of mRNA molecules in the cytoplasm is a key to determining protein synthesis. Eukaryotic mRNA is more long-lived than prokaryotic mRNA. Nucleotide sequences that influence the lifespan of mRNA in eukaryotes reside in the untranslated region at the 3' end of the molecule. After translation, the polypeptides are going to undergo processing. This includes cleavage and chemical modifications. The length of time each protein functions is actually regulated as well by selective degradation. So not only does the protein need to be made and has to undergo processing, but how long the protein is able to function is also regulated as well. Cells mark proteins for degradation by attaching ubiquitin to them. This mark of ubiquitin is recognized by proteasomes, enzymes that are going to be able to recognize and degrade the proteins. Only a small fraction of DNA codes for proteins, and a very small fraction of the non-protein coding DNA consists of genes for RNA, such as rRNA, ribosomal RNA, and tRNA, that transfer RNA, both of those needed for translation to happen. In the past, genes that did not encode a protein product or have a known functional RNA, they were considered junk DNA. However, there have been many genomic studies that have now cast doubt on this description. A significant amount of the genome may be transcribed into non-coding RNAs, which are referred to as ncRNAs. Non-coding RNAs regulate gene expression at 2 points: mRNA translation and chromatin configuration. MicroRNAs, or miRNAs, are small, single-stranded RNA molecules that can bind to mRNA. These can actually degrade the target mRNA or block its translation. It is estimated that expression of at least half of all human genes may be regulated by these miRNAs. What happens here is that the miRNA, this small RNA protein complex, which is bound to a protein itself, can recognize a portion of the target mRNA. When it binds to it, it either degrades it or it blocks it from being able to bind to a ribosome in order for translation to happen. Small interfering RNAs, or siRNAs, are similar to miRNAs in size and function. The blocking of gene expression by siRNAs is called RNA interference, or abbreviated RNAi with the little I. RNAi is used in the laboratory as a means of disabling genes to investigate what their function is. Embryonic development is perhaps the ultimate example of precisely regulated gene expression. During embryonic development, a fertilized egg gives rise to many different cell types. Cell types are organized successively into tissues, organs, organ systems, and the whole organism. Gene expression orchestrates the developmental programs of animals. The transformation from zygote to adult results from cell division, cell differentiation, and morphogenesis. Cell differentiation is the process by which cells become specialized in structure and function. The physical processes that give an organism its shape constitute morphogenesis. An egg's cytoplasm contains RNA, proteins, and other substances that are distributed unevenly in the unfertilized egg. Cytoplasmic determinants are maternal substances that are in the egg, and these influence early development. As the zygote divides by mitosis, cells contain different cytoplasmic determinants which lead to different gene expression. The other important source of developmental information is the environment around the cell, especially signals from nearby embryonic cells. In the process called induction, signal molecules from the embryonic cells cause transcriptional changes in nearby target cells. Thus, interactions between cells induce differentiation of specialized cell types. Differential gene expression results from genes being regulated differently in each cell type. Materials in the egg set up gene regulation that is carried out as the cells divide. The gene regulation systems that go wrong during cancer are the very same systems that are involved in embryonic development. Mutations that alter normal cell growth and division can lead to cancer. Oncogenes are mutations in genes called proto-oncogenes. So normally the genes are called proto-oncogenes, and these genes code for proteins that stimulate normal cell growth and division. Mutations that happen in these genes are called oncogenes. An oncogene arises from a change that either increases the amount of the proto-oncogene's product or in the activity of the protein. The genetic changes that convert proto-oncogenes to oncogenes fall into four main categories: epigenetic changes, translocations, gene amplification, and point mutations. These are genetic mutations that can turn proto-oncogenes, which again are normal genes that code for proteins that are involved in stimulating the normal cell growth and division. And these are genetic changes that can turn those proto-oncogenes into oncogenes, which can cause cancer. The first one is epigenetic changes, where there's a mutation in a gene for a chromatin modifying enzyme that can lead to the loosening of the chromatin and then inappropriate expression of a proto-oncogene, so it gets synthesized more in excess. Translocation can also happen where the gene is moved to a new locus under new controls, and this also can allow the protein to be synthesized in excess. There can be gene amplification where there ends up being multiple copies of the gene, also again allowing that gene to be in excess. And then there can be a point mutation within a control element, or even a point mutation within the gene. A point mutation within a control element can again allow the protein to be made in excess, and a point mutation within the gene itself can make a hyperactive or a degradation resistant protein that allows it to stay around longer. All of this is going to lead to inappropriate cell growth. Tumor suppressor genes are another type of gene that are involved in cell growth and proliferation. These specific genes normally help prevent uncontrolled cell growth. Mutations that decrease these protein products of tumor suppressor genes may contribute to cancer onset. Tumor suppressor proteins normally repair damaged DNA, control cell adhesion, and act in cell signaling pathways that inhibit the cell cycle. So these genes make sure that the cells don't grow one on top of another in an uncontrolled manner. Mutations in the RAS proto-oncogene and a p53 tumor suppressor gene are actually common in human cancers. So we see a proto-oncogene and a tumor suppressor gene having mutations. Mutations in the RAS gene can lead to the production of a hyperactive RAS protein and increase cell division. So this is going to allow cell division to increase, and without that tumor suppressor gene working like it should, not only does cell division increase, but that allows the cells to grow on top of one another, touch each other, things that normally wouldn't happen if these controls were in place. Suppression of the cell cycle can be important in the case of damage to a cell's DNA. p53 prevents a cell from passing on mutations due to DNA damage. However, if there are mutations in the p53 gene, then that is going to prevent suppression of the cell cycle. If DNA is irreparable, if the DNA cannot be repaired, then p53 would activate cell suicide. So here's an example of what happens. If UV light comes in and damages the DNA, because UV light is one of the ways that DNA can be damaged, and we see that in both these figures, and p53 is active, what's going to happen here is that DNA damage is going to allow for an intracellular signal to be formed, which are these protein kinases. These protein kinases are going to activate p53, and then p53 is going to promote the transcription of a protein that is going to inhibit the cell cycle. So when p53 is present and can be activated, it is going to inhibit that DNA from being replicated, not allowing that damaged DNA to be replicated. But if there's a mutation in p53 that would allow it to bypass this mechanism, and it would allow that mutated DNA to go forth still and allow that DNA to be replicated, allowing the cells to be replicated as well with that faulty DNA. Multiple mutations are generally needed for full-fledged cancer. This then increases with age. At the DNA level, a cancerous cell is usually characterized by at least one active oncogene and the mutation of several tumor suppressor genes. So it's not just one, but a combination of these. Routine screening for some cancers, such as colorectal cancer, is recommended. In such cases, any suspicious polyps may be removed before cancer can progress. Breast cancer is a heterogeneous disease that is the most common form of cancer in women in the United States. A genomics approach to profiling breast tumors has actually identified 4 major types of breast cancer. Individuals can inherit oncogenes or mutant alleles of tumor suppressor genes. Inherited mutations in the tumor suppressor gene adenomatous polyposis coli are common in individuals with colorectal cancer. Mutations in the BRCA1 or BRCA2 gene are found in at least half of inherited breast cancers, and tests using DNA sequencing can detect these mutations. A number of tumor viruses can also cause cancer in humans and animals. Viruses can interfere with normal gene regulation in several ways if those viruses are actually able to integrate into the DNA of a cell, and some can. Viruses are powerful biological agents, and we will be discussing them in more detail in the next video.